Hello everyone, thank you very much for joining this poster session. I'm Paul and today I will present a toolkit for concretely describing non-canonical polymers and complexes, and we call this toolkit BpForms and BcForms. But before I get into that, let me thank my fantastic colleagues for contributing to this work. Our motivation for creating BpForms and BcForms was that we wanted to create whole cell models. And as we know, in cells we not only find canonical polymers, we also find modified polymers, and we need to describe them. There were already formats out there, but molecular formats had the issue that they poorly scaled to macromolecules, and formats for macromolecules were typically chemically ambiguous. So we set out to create a format that concretely describes chemical structures, abstracts away the molecular details, and is machine and human readable. And on the next slides, I'd like to show you the idea of this format. And I start at the very low level with a single residue, selenocysteine. This selenocysteine can be described in its structure by its SMILE string. And to the SMILE string, we associate a unique identifier. Here it's U. We can do this for different residues as well, and we can put them in a sequence similar to what FASTA does. But where BpForms goes a little further is that BpForms already exactly describes how the different residues are bonded to each other. And to do that, every residue has a left bond atom, left displaced atoms, a right bond atom, and right displaced atoms. And then we know exactly what the bond looks like, and we form a peptide. We can have multiple peptides, and peptides can have residues like cysteine that can form crosslinks. And in order to describe the crosslinks, again, we need to describe the bond atom and the displaced atom. So here we have now a dimer, and it's important to note that within this dimer, every atom has its unique coordinate. There's the atom coordinate in a residue, then there's the residue coordinate in a sequence, and there's the subunit coordinate in a complex. So that's basically the idea of BpForms, but we had to put this into a machine and human readable string format. And how we did this is shown here. For instance, we have single letter residues and we have multiple letter residues, where multiple letter residues are encoded in braces. We also see that if you don't find the residue of your interest in our predefined alphabet, you can define it yourself using square brackets. You just need to make sure that you describe the structure and also left bond atom, right bond atoms, and several displaced atoms. What else do we have in this string? We see that we can specify crosslinks using the ontology of crosslinks. So here we say that we have a disulfide crosslink, and for that we have already defined what the left bond atoms, right bond atoms, so what the bond atoms are and what the displaced atoms are. If you need to describe further crosslinks, you can do so again using this user-defined format with the square brackets. All right, once you have defined your polymers, you can put them into a complex. And within this complex, you might have, again, crosslinks, which you can describe using the ontology or the user-defined form. Just to give you an idea of what's already in our alphabet, we have several hundred residues for DNA, RNA, and protein. And we also have 36 protein crosslinks in our ontology of crosslinks. So once you have defined, again, polymers or complexes, you might want to perform certain computations on them. And you can do this using our user interfaces, namely the web app, the command line interface, the REST API, and the Python API. And you can calculate things like, say, the molecular weight at a certain pH value. You can also put BpForms strings into network standards. For instance, you can put it into CellML and SBML. And we believe, especially by putting this into these network formats, BpForms allows you to improve, expand, and merge models. And the reason why we believe this is basically shown in this figure here. So what you can see is that we have the MAP kinase cascade model, but the species have rather generic names. So it's not quite clear what this actually means. But you can annotate these species using BpForms. And what you do is you also annotate where exactly the phosphates are located. And you might figure out that you have left out a certain species and a certain reaction. So you can extend your model by these species and reactions. If other users were also to use BpForms, it would be easier to merge models with other models. And through this merging, you may figure out that there are new proteoforms or new reactions that you discover. BpForms, like I said earlier, has atomic resolution, so it allows you to check whether or not your reactions are actually mass balanced. If they're not, like in this case, you can correct for that. So basically, you end up with a much more comprehensive and detailed model. If you want to give BpForms a try, probably the best starting point is to go to our website, bpforms.org. But you can also have a look at our GitHub repo or interact with our REST API. All right, to wrap things up, BpForms precisely describes non-canonical macromolecules. It supports the abstraction needed for network scale research. It facilitates quality control, exchange, and integration of post-genomic data. And it comes with a user-friendly web app, REST API, command line interface, and Python API. With that, I'd like to thank you for your attention, and I look forward to responding to your questions in the chat.